Hi, everybody. Today we are going to be focusing on the muscular system. You may know that we'll have three categories of muscles: a skeletal, cardiac, and a smooth muscle. The rest of this lecture is going to be focused on the skeletal muscle as being the most common one. We will focus on how the muscle contraction takes place for this, look at some of the anatomical features in terms of both macroscopic and microscopic anatomy. And do note that if you feel comfortable with the skeletal muscle and its contraction, it would be very similar to what you will learn later for the cardiac muscle as well. So looking at the characteristics of these muscles, there's three shapes that are associated with these muscles. When you're looking at the skeletal muscle, we usually depict the skeletal muscle as kind of the cylindrical structures. So these muscles can be pretty long, and therefore they can be referred to as muscle fibers. So this is a typical way of depicting your skeletal muscle. They also have these patterns of lines going through them, which is referred to as striations. They do have multiple nuclei. One of the main reasons because they are long and they need the basically multiple control centers. The type of movement they create is voluntary. Again, looking back at what you learn about the nervous system, the somatic branches of the nervous system was responsible for controlling a skeletal muscle movement, which was voluntary. Because of the length of these cells, again, they are elongated. They call myofibers. The word myo implies muscle, and fibers implies that these cells are long. Also contains what we consider glycosomes. Glycosomes is where you can store glycogen. Glycogen is, if you guys recall, is a complex carb. And basically, this is the one that is going to be responsible for a storage of nutrients or your sugar, quote unquote, for the muscle contraction. You also have what we consider myoglobin. Myoglobin is a version of hemoglobin. So hemoglobin is the protein in your blood that stores CO2. Stores O2, not CO2. Now we have a similar molecule called myoglobin. Again, myo implies muscle, and that helps you store oxygen inside the muscle cell. And it is separated from hemoglobin. So basically, hemoglobin brings the oxygen to your muscles, and that oxygen gets transported from the hemoglobin to the myoglobin for muscle usage. Characteristic of the cardiac muscle, best described as it is branched. You still will see a striation pattern on these muscles, so very similar again to the skeletal muscle. They are referred to as cardiocytes, and their movement is involuntary. Again, kind of makes sense, and they're not under our control. A smooth muscle, as is depicted here, they are spindle shape or fusiform, which basically means thick at the center, and as you go on either ends, they narrow down. As you can see on the picture, there is no striation pattern that is depicted in a skeletal or cardiac muscle, and the movement created by the smooth muscles are identified as involuntary. Looking at some of the locations you can find these muscles, you have it in skeletal muscles are basically anything that's attached to your skeletal system. Cardiac is anything that is associated with your heart, and a smooth muscle can be found in your stomach, your respiratory system, blood vessels, uterus. So they're typically found lining many of the hollow organs in our body. Now let's go ahead and take a look at some of the characteristics of the skeletal muscle. So kind of the first one is logical, produce a skeletal movement. So again, as the name implies, these muscles are attached to a skeletal system, and with the help of joints and ligaments and tendons, it allows the movement of the skeletal system. It helps you maintain your postures. It protects soft tissue, for instance, the abdominal muscles. There are four layers of that, and these muscles are basically going to be protecting the internal organs, typically the digestive system. They guard the entrances. These sphincters, these circular muscles, can actually be both a skeletal, which basically means we have control over them, or they can be a smooth muscle. However, the ones that we're concerned about for this lecture is the skeletal muscle, which again gives us voluntary control over exits of the body. For instance, you have that at the urethra, at the end of the urinary system, as well as the end of the digestive system connecting to the anus. And a big factor, skeletal muscle play a big factor or role in maintaining your body temperature, or in another sense, maintaining your core body temperature at that 37 degrees centigrade. Some of their main characteristics for the muscles include excitability, as they are ability to respond to a stimuli. This is stimuli typically associated with your nervous system, which we already talked about in terms of, again, somatic branch of the nervous system. They do contract, so they do have the ability to be shortened when they are stimulated. And they also can get elongated. Basically, you can stretch the muscle up to a certain point, but after that point, you basically are going to have a difficult, your muscles literally start to rip apart. But there is a range, as you have the normal range, and then you can contract the muscle to make it shorter, or start to stretch the muscle that makes it longer. The extensibility is not optimal in many cases, again, because that's not something that your muscle needs to undergo, but up to a certain level, it can deal with it. And then lastly is elasticity. So again, if you contract or basically do any changes in the muscle will go back to its original shape, and then it has the recoiling ability to go reset back to its original length. Now, the original cells we start in the muscles are referred to as the myoblast. These myoblasts are basically, as you already learned, the word blast means basically immature cells. Myo means muscle. Now, these cells start to actually come from an embryonic stem cells associated with mesoderm. These cells eventually differentiate to form your myoblast. And myoblasts, as you can see, there are individual cells with individual nuclei. But as they start to develop, what happens is that multiple myoblasts are going to join each other and form what we identify right here as a myotube. Myotube is an immature structure which will eventually become your skeletal muscle. So if I ask you guys, how does a skeletal muscle end up with multiple nuclei? It's the result of multiple myoblasts merging to each other to form a myotube and eventually the skeletal muscle. Fascinating thing about the skeletal muscle is within the first 7 weeks of embryonic development, you will have muscles that have the capacity to contract, which is absolutely amazing. Now, when you look at the skeletal muscle, there are 3 layers of connective tissue that can be found in the muscles. Starting with the outermost layer, if this is my whole muscle that connects basically to a bone, let's say this muscle is your, again, I'm just gonna give you a common name, bicep brachii, which is your arm muscle. So you have a connective tissue that is wrapped around this whole muscle, and it's called the epimysium or epimysium. Now, this whole muscle is made up of smaller units. They pulled one of them out right here. This smaller unit as a whole is referred to as a fascicle. Notice again, fascicle has a layer wrapped around it as well, and that layer is referred to as the perimysium. Furthermore, if you look inside the fascicle, which is this larger structure, you have a smaller structure like what I've drawn here or what they pulled out right here. These are identified as an individual muscle fiber, which we also can call myofibers. Again, myo implies muscle. And the connective tissue you see wrapped around right here, this connective tissue is called the endomysium or endomysium. So 3 levels or 3 layers of connective tissue within your muscle. To kind of just depicting it for you in a little bit more detail or simplified version of what I have here. Sorry guys, 1 second. Oops, that's not what I wanted. So if this is my whole muscle, so this is whole muscle, like again, your biceps brachii, and the connective tissue, which is here, would be your epimysium or epimysium. Within that, you have these circles, and again, these circles take over all of these in space. These circles are your fascicles, and the connective tissue wrapped around them would be perimysium. And then within that, you will have other structures, which are these individuals that you see right here. These are individual muscle fibers, and the connective tissue wrapped around that would be endomysium. So all I've done is basically depicting the picture as a cross section versus this one, they're showing you an actual muscle pulling the structure out. So I just showed you a cross section of your muscle. Now, when we look at the muscle fiber, looking at some of the microanatomy of the muscle fiber and vocabulary associated with it. First, we have sarcolemma. Sarcolemma should remind you of, I should first of all start by saying that the word sarco also implies muscle. So sarcolemma is basically the plasma membrane that is found around your muscle fiber. Not to be mistaken with the endomysium, which is a connective tissue that sits on top of the sarcolemma. Sarcoplasm should remind you of the word cytoplasm, which is basically space inside the cell for just the muscle cell. You have a specific structures inside your muscle fiber called myofibril. These are densely packed, rod-shaped structures, and they're found again inside a muscle fiber. They're extremely important in terms of your muscle contraction. What we also have is a modified version of your endoplasmic reticulum, which we learned earlier being a main organelle inside your cell. We shorthand endoplasmic reticulum to ER. But in case of muscles, we specifically call them sarcoplasmic reticulum. The importance of sarcoplasmic reticulum is that it stores calcium that is needed for muscle contraction. Lastly, when we think about a muscle cell or muscle fiber, as we learned earlier, they respond to a stimuli, which is basically coming from your nervous system. Now, the electrical impulses that are coming from the nervous system need to have a way to first be passed down to my muscle fiber, and then eventually spread through the muscle fiber. So these T tubules, which are invagination of my plasma membrane sarcolemma, allows the fast depolarization, which is the same thing as your action potential or electrical impulses of my muscle fiber. So basically what we're saying here is that your nervous system communicates with the skeletal system, the skeletal muscle system. The electrical impulses gets passed down from the muscle, from the nerve to the muscle, and then it spreads throughout the muscle fiber using your T tubules. They do have, these T tubules have a special cross-sectioning or cross channels referred to as a terminal cisternae. And they are found, these terminal cisternae, or occur on either side of my T tubules. So let's take a look at this. And I'll show you guys one at a time. So the picture you see right here, this large cylindrical structure you see, you can identify nuclei here. So this whole structure is your muscle fiber, an individual muscle fiber. What has been peeled off is your plasma membrane, which is your sarcolemma. What they're pulling out, these rod-shaped structures that are being pulled, are your myofibril, as we mentioned earlier, right? So sarcolemma is the plasma membrane. Myofibril are the rods inside the muscle fiber. And as I said, they are important in terms of muscle contraction. Please do note that the muscles in general do carry a lot of mitochondria. And if you guys recall, mitochondria is basically how you produce your ATP. Because muscle contraction requires a significant amount of ATP, then you also do have a lot of mitochondria present. We also learned that you also have a significant amount of complex carbohydrates, which are stored in glycosomes. And we also have significant storage of oxygen by having myoglobin present inside my cell. Again, same picture, now you're kind of zooming in. Each of the cylindrical structures you see right here are representation of a myofibril. You can see your mitochondria again, kind of stashed between these myofibrils. What you see in blue is your sarcoplasmic reticulum, which stores significant amount of calcium. And you please do note how it's wrapped around your myofibrils. And the light blue color that you see kind of going again throughout your muscle cell, and it's a continuation of your sarcolemma. Here's your sarcolemma, here's the continuation. These light blue color are your T tubules, which are responsible for spreading the electrical impulses or depolarization within your muscle fiber. Now, why do I care about myofibrils? Myofibrils account for about 80% of your muscle cell. There are literally thousands of these inside a muscle cell. One of the main features of myofibrils is that it creates the striation pattern we see in terms of your muscle. The myofibrils are broken down to individual units, vertical units called sarcomeres, which are my contractile units. And the sarcomeres are contain two main proteins called actin and myosin. Myosin being my thick filament and actin being my thin filament. So I know I gave you guys a lot of information. So what I'm gonna do right now is just do a quick drawing for you guys to show you what all of these structure actually looks like. So what we're gonna start with, this is a simple depiction of what we have talked about. Let me change my pen color. So we're gonna start with looking at a muscle fiber, which is my cell, right? So this is my muscle fiber. The membrane that you see right here would be my sarcolemma, packed inside my muscle fiber. I have thousands of these cylindrical units, which are called myofibrils. You guys can pronounce it differently. So I'm trying to color code so you guys can follow. These are myofibrils. The myofibrils are broken down to smaller units, vertical unit breakdown for my muscle. Each of these units, each of these new branching of the myofibril are called sarcomere, with the goal of basically contraction. So these are your contractile units. What can I find inside my sarcomere are two things. The thick filaments, which are myosin, and I'm only going to draw a few of them, but assume it's repeated for all of these. So these ones are my myosin, which is also called the thick filament. And we also have what we identified as thin filaments, which are basically these lines that I'm drawing. And again, you can see some overlapping with your green color. Okay, so these are my actin, and they're called thin filament. So if we're starting from the smallest to the largest, kind of working and looking at this whole idea from the smallest to the largest, kind of write this down. You have actin and myosin, which forms units called sarcomere, right? Sarcomere is packed inside myofibrils. Myofibrils are packed inside myofibers. My pen has my own mind, which is basically a muscle cell. Muscle cells are packed inside fascicles, and fascicles are packed inside my whole muscle. So basically, you're going from the smallest structure, which is actin and myosin, to the largest structure, which is your whole muscle. Now, going into a little bit more detail about this is looking at some of the features of your myofibrils. So each of the sarcomeres have regions are called A-band, which are the dark regions, typically associated with myosin. You have H-zone, which are regions that are lighter in the middle of the dark A-band. And again, I'll show you guys in just a second what these are. M-line, that's basically bisects or divides your H-zone right in the middle. I-band, which is the lighter region, and then Z-disc, which are basically separating each of the sarcomeres from one another. So let's take a look at what they look like if I was drawing them. So let's say I'm focusing on a myofibril, right? And we know myofibrils are made up of sarcomeres. So I'm going to draw two sarcomeres next to each other. So here is one sarcomere, again from here to here. And here is my second sarcomere. Now, what do we have inside the sarcomere? Is we have the two proteins. So the big thick protein that we have, and we identified that earlier, that's the thick filament, which is your myosin. And you have your thin filament, which are basically the skinnier or the thinner lines are called actin. So here is my basically depiction of actin for the two neighboring sarcomere. okay? So again, the green section is myosin, and the thin lines are actin. And the black line that are separating this is basically marking my two sarcomeres. So let's go ahead and take a look at the vocabulary for these. Now, anytime you see these lines that are basically bordering my each sarcomere, these are identified as my Z disc. So Z disc is basically a marker for the end and beginning of each sarcomere. Regions where I only have actin, so from here to here, from here all the way to here, and from here to here, these regions are called the I band. And again, these are only and only actin, or my thin filament. The regions where it's the length of the myosin, basically from here to here, right smack between the two I bands, from here to here, these are called the A band. And again, notice that the A band, which are basically the length of the myosin, also includes a little bit overlapping actin, right? So see overlapping actins. So overlapping actins and the length of the myosin would be my A band. Now, the midsection, I'm going to change my color so you guys can see it a little bit better maybe. The midsection of the A band, which is from here to here, where there is no actin overlap and you only have myosin. So from here to here and from here to here, these two regions are called the H zone. So here is one H zone, here is another H zone. And then running right in the middle of the H zone, the dashed line that I'm drawing right here would be representation of my M line. So if I ask you what is sarcomere made up of, you will say actin and myosin. If you're looking at the breakdown of that, we have the I band, which is only actin or thin filaments, A band, which is the myosin and its overlapping actin, H zone, which is the midsection of your myosin, where you only have myosin and no actin overlap. And the line that runs right in the middle of the H zone is called the M line. Now, we have talked about striation before, right? Striation being the light and dark pattern of the muscle you see. So in this case, if you kind of think about it, if the light is hitting this sample from behind and you guys are looking at it through the microscope, where you have actin, because these are thin filaments, lots of light can pass through, and therefore these regions appear as light. However, the part that is associated with myosin, because myosin is thick, it absorbs a lot of light, and therefore the light coming to your eyes will be limited. So these regions, which is basically the length of your myosin, will appear as dark. So when you look at it under the microscope, you get light, dark, light, dark, light, and then you guys again can repeat it over and over again, and that gives you the striation pattern you see in a skeletal muscle. So again, if I kind of highlight the same point for you in writing, the dark band, write it this way. Dark band is going to be your A band, which is myosin, right? And your light band is going to be your I band, which is only actin. So here you can see the depiction again. Here is my Z disc. The darker red lines would be my myosin, which is also depicted as the A band. Regions where I only have the blue lines that are my actin, I identify as the I band. And this midsection right here, when there's only myosin and no actin overlap, that would be identified as my H zone. Now, again, actin and myosin, kind of giving you a little more information. They are very orderly matter. Actin filaments are thin, we already know that. They extend across your I band, right? Which we identified I band being your actin, and partly through your A band, which is the overlapping region between the A and A band the overlapping region between actin and myosin. And they're anchored in the Z disc. Again, it kind of gives you the view of where the end and the beginning of each of the sarcomeres are. Myosin filaments or myofilaments are the thick filaments. They extended through the length of the A band, which we already established on our picture. And they're connected at the center to the M line, which also runs right in the middle of the H zone. Now, if you kind of think about this whole concept, we have an understanding that actin and myosin are going to cause my muscle contraction, because that's basically your sarcomere, and sarcomere is the contractile unit of your muscle. So the question is, how does a muscle contraction take place? So imagine your myosin and imagine your actin. So first thing I want you to understand is, when you look at a myosin, which is your thick filaments, coming off of your thick filaments, you have these kind of extensions coming off, which are identified as myosin head. Now, myosin head wants to interact with the actin, and the way it interacts it, if you think about the blue line being your actin, it's made up of individual subunits, and these subunits have this little gap inside of them. And these little gaps right here, this little indentation, are identified as the myosin binding site of actin. So basically, when you look at muscle contraction, myosin heads interact with the myosin binding site of actin. And these heads, notice there's these tiny arrows, these heads are moving in, which basically means it takes the actin and pulls the actin inside the myosin. So actin during muscle contraction overlaps with myosin at the higher level. So kind of understand this, and I'm gonna redraw this in a very fast drawing so kind of you guys can understand it. Here is my myosin, here is my actins, right? So these have heads, these would have the myosin binding sites. So when these heads interact with the actin, the heads are going to create this motion, kind of like when you row, okay? And as they create this motion, they're gonna take this actin and move it in toward the M line, which is sitting right in the middle of your myosin. Same thing with this one. Heads are right here. So when they move, they're gonna push your actin inward into your myosin. So when your muscle is relaxed, the overlap between your actin and myosin is present, but it's not as much. When your muscle is contracting, the interaction between the myosin binding site of your actin and the myosin heads of your myosin causes the actin to move into your myosin, increase the overlap, and causes the whole sarcomere to shorten. So when you think about your muscle contraction, it's usually depicted as muscle getting shorter, which is exactly what we're describing here. Now, if you're looking at this whole concept, again, going back to the I band and A band before we move forward, the I band during muscle contraction is going to get shorter because I band gets pushed into the myosin. So the regions which is only actin right here will be moving into the myosin, so these regions get shorter. So again, if I kind of draw this whole picture again during muscle contraction, you will have this scenario. So notice right here, the overlap is small. Right here, the overlap is great. So in this case, let's say this was the actual length, right? So here is my I-band right here. Notice the I-band got shorter. A-band, which was the length of my myosin, is still gonna be the same because nothing is happening to my myosin. All I'm doing is increasing the overlap. So it is still the same length of the myosin. So A-band remains the same. H-zone, however, which was only and only your myosin region, which is right here. So this is my H-zone, correct? But I only have myosin. Look at my H-zone now. It got shorter, right? So H-zone is longer during muscle relaxation. H-zone becomes shorter during muscle contraction. Again, A-band remains the same during both muscle contraction and relaxation. And your I-band gets shortened as the muscle contracts because it increases its overlap with myosin. Now here's the thing. If you're describing here the process of muscle contraction in a very simple, basic manner, what is preventing every single muscle in your body to contract every single second of every day? And that driving force, what prevents it is two regulatory proteins that are found around the actin, covering your myosin binding site. And these two proteins are the longest string that you see in orange called tropomyosin, and the little protein subunits in yellow called troponin. So under the normal condition when your muscle is relaxed, the troponin and tropomyosin are covering myosin binding sites on your actin, which is the blue line, and preventing actin to interact with the myosin heads. And this basically keeps your muscle relaxed. Now, what causes muscle to contract is presence of calcium, which I'll talk about in detail as we go through the PowerPoint. As calcium enters your cytoplasm or sarcoplasm more specifically, it's going to interact with troponin, the yellow dots, and moves troponin. Troponin is covered with tropomyosin. So as troponin is moved, tropomyosin also moves along. And that causes the little myosin binding site to free up, which allows the muscle to contract. So one more time, when the muscles are relaxed, the covering, myosin binding site is covered by troponin and tropomyosin. When you have calcium present inside your sarcoplasm, calcium binds to troponin, which moves it gently. Troponin movement causes tropomyosin to move as well because they're connected, which allows the myosin binding site of the actin to free up. In such scenario, the actin can interact with the myosin heads, and that causes the motion we're depicting right here, and allows the muscle to contract and get shorter. Now I'm going to stop here and we'll continue with the second part of focusing on a skeletal muscle contraction on our next presentation.